Hello everyone, let's see about the techniques for crop improvement in banana and papaya. Crop improvement in banana. Banana is one of the major food crops grown in India. In respect of area and production, it ranks second only to mango in this country. The banana culture in India is as old as the Indian civilization. Breeding banana is almost a difficult exercise due to complexities resulting from parthenocarpy, sterility, polyploidy, and vegetative propagation. The uniqueness lies in the fact that in banana, which is almost sterile, raising sexual progeny in sufficient numbers to combine desirable characters and at the same time resulting in another sterile plant is indeed very difficult. Breeding methods of banana. They are of three types. They are hybridization, mutation breeding, biotechnological methods. Hybridization. In India, breeding work was started at Central Banana Research Station, Adurai in 1949. Hybridization technique of banana is different from other crops. Pollination is carried out in the morning. The bunches of female plant are bagged at shooting and each successive hand is pollinated as it is exposed. At ripening and maturity, the bunch is cut and the seeds are extracted. Evolution of hybrid progenies from seedlings to harvest may not be the correct phase. Instead, evolution of same under next vegetative phase, that is, sucker to harvest stage, will be ideal as full expression of field potential could be observed only in the second crop of F1 progeny. The first crop takes more than 15 to 19 months, where most of the energy of the plants is needed for comb formation. Bud mutation breeding. Bud mutation in Indian banana is very common, perhaps due to spontaneous rearrangement of chromosomes in somatic meristem and structural reassignment. A great majority of edible bananas are diploids, a condition that interferes with normal equilibrium of plant and may provide the requisite stimulus to structural rearrangement of chromosomes, leading ultimately to the evolution of new gene complex. Biotechnological methods. Plant tissue culture and molecular biology techniques are applied to enhance the handling and improvement of banana. Important applications of cell biology are micropropagation for rapid multiplication and germplasm exchange, embryo culture or rescue for in vitro seed germination, cryopreservation at germplasm, and genome manipulation through genetic engineering using cell suspension or protoplast culture. Although Wilstick et al. Reported that somoclonal variation through micropropagation is of limited use in plantation breeding. It has been successfully applied in Taiwan for the development of improved Cavendish banana cultivars with resistance to fusarium wilt and acceptable fruit quality. Next one is the crop improvement in papaya. In papaya, breeding methods are of four types. They are inbreeding and selection, hybridization, heterosis breeding, and mutation breeding. Inbreeding and selection. In dioecious lines, suitable male plants are selected from same progeny which have resemblance to female plants in vegetative characters such as stem and leaf color, stem thickness and height at flowering, etc. Progenies raised from seed inbreeds are screened and decided male and female plants are selected for further submating. That is, crossing between the female plant and male plant of the same cultivar. The process is to be continued for 7 to 8 generations to achieve uniformity of a group of characters. In this method, the progeny will have male and female in equal population. Many dioecious cultivars have been bred by this method. As a result of inbreeding and selection for 8 generations during 1966 to 1982, the following varieties were developed. They are the Pusa delicious, gyno-dioecious line, heavy yield, fruit very sweet in root and good flavor, medium tall plant. Pusa majesty. This is the gyno-dioecious line plant, high papain yielder, better keeping quality fruit, tolerance to virus and nematode. Hybridization technique. First one is using gyno-dioecious lines. It involves selfing regular and prolific bearing hermaphrodite or crossing the female with the hermaphrodite. Suitable hermaphrodite plants which do not vary with the climatic changes are selected. Of the various types of the flower produced by the hermaphrodite plants, elongata and pentandra types are selected for selfing. Selfing is to be continued with selected hermaphrodite plants for at least 3 generations for uniformity of characters. In the case of female and hermaphrodite plants, submating between the said types of female plants are selected and submitted with hermaphrodite plant. Seedling raised from S1 hybrid inbreed is screened and the said female and hermaphrodite plants are selected for further submating. The process is to be continued for 7 to 8 generations till homozygosity is achieved. Using dioecious lines. It has been established that female plants are more productive than hermaphrodite ones. Due to crossing, most of the cultivars are highly variable. Hence, it is considered appropriate to submit the selected female and male plants so as to bring the homozygosity. Hence, suitable male plants are selected from the same progeny which has the same resemblance to female plants in vegetative characters such as stem and leaf color, stem thickness and height at flowering, etc. Progeny is raised from Inbreeds are screened and the desired male and female plants are selected for further submating. This process is to be continued for 7 to 8 generations to achieve uniformity of the group of characters. Heterosis breeding. At IIHR Bangalore, an F1 hybrid, namely Surya, was released recently. It is gynodioecious in nature and produces about 75 to 80 fruits of medium size, weighing about 600 to 800 grams. The flesh is red in color, firm, sweet to taste with the traces of 14-degree bricks. A dwarf mutant was isolated in generation by treating the seeds with comores. Repeated submating among the dwarf plants and had resulted in the establishment of homozygous dwarf line named as Busan Anha. The first bearing height was about 106 cm from the ground level. Recent breeding scheme, the papaya, Drew 2016 stated to improve plant through conventional breeding, marker, assisted breeding, genetic modification. Moreover, they developed segregation population for future breeding work, which will facilitate future research and increase selection of new commercial varieties. They followed the following strategy in improving papaya breeding. It includes the following methods: plant and grow several lines of papaya to maturity, identified important trait for papaya breeding program, evaluated and traits of interest in all the lines. On first harvest, the collected data of the selected trees bearing fruit having best consuming quality cross-pollinated and selected trees with commercial varieties with the view to get segregated population with improved eating quality. On second harvest, they collected data on correlation and studied the repeatability. On third harvest, they select 2 trees for each trait with a high difference and finally cross. Thank you.